hear me? Okay. First, I would like to thank very much the organizers for having me and allowing me to come speak to you guys. I am a third-year PhD student, and I'm going to tell a story that is relatively small in scope. I hope to convince you here today that long non-coding RNAs, lncRNAs, are important members of coding gene networks. And I would like to also convince you that this is based on the property that some long non-coding RNAs have, which is propensity to form sense-antisense pairs, SAS pairs. So I'm going to start out with a short introduction, probably unneeded, about long non-coding RNAs and sense-antisense pairs. Long non-coding RNAs are structurally identical to mRNAs. They're just not translated. This is a schematic of a long non-coding RNA, the non-coding RNA that pairs with BDNF. It's part of a genomic structure which produces two transcripts that have shared sequence on opposite strands. That's this SAS sequence. This transcript here is BDNF. The key point about sense-antisense overlap is that it is exonic shared sequence, and this creates two transcripts that have a reverse complementarity. So this is a table which shows some long non-coding RNAs in sense-antisense pairs and why we think that sense-antisense pairs can be regulatory. Long non-coding RNAs in sense-antisense pairs can regulate the coding gene transcriptionally, epigenetically, or post-transcriptionally. And this potentially regulatory interaction can occur in the nucleus, or it can occur in the cytosol as a result of the sense-antisense being exonic. So I'm going to show you two data sets today. The first data set comes from an experiment where we tried to test the hypothesis that long non-coding RNAs may be differentially regulated in a cancer model. The cancer model that we used was MCF7 cells treated with estrogen. This mimics malignant transformation in a breast cancer model. We found 127 long non-coding RNAs that were differentially regulated compared with controls. And when we started investigating this group of long non-coding RNAs a little closer, we noticed that some of them, about 60% of them, were in these sense-antisense overlaps with coding genes. When we looked closer at these coding genes, we saw that often they were genes that could be possibly part of cancer networks. So this is one data set that, or one data point that came out of this study. This is a long non-coding RNA, BC03, this BC here. It is in it's in a cluster of sense-antisense overlaps with coding genes. 3 of the coding genes in this sense-antisense cluster are known to play a role in cancer progression. And further corroborating that this is an estrogen-responsive long non-coding RNA, we found an ER-alpha binding site through ChIP-seq data from ENCODE that's publicly available in UCSC. So the second data set that I'm going to draw on today is a data set where we looked not just for long non-coding RNAs, but we also looked for differentially expressed sense-antisense pairs in another cancer model, or another cancer, this time melanoma. This was not a model. These are actual melanocytes in melanoma cells. And this was not a microarray, as in the breast cancer work. This used Illumina high-seq data. So in this data set, we found 2,619 sense-antisense pairs that were differentially regulated in cancer versus controls. And we quantitated both the expression of both transcripts, not the pair as an entire region, but it is 2 separate transcripts. So this is one data point that came out of this experiment. This is the coding gene MAO. It is a known tumor suppressor. As you can see, it is downregulated in cancer. It shares sense-antisense with a long non-coding RNA overlapping the 5' end. And this long non-coding RNA is also downregulated in cancer. Interestingly, this coding gene is known to be repressed in cancer based on promoter hypermethylation. And as I showed you before in that table, epigenetic regulation of the coding gene is one possible mechanism for sense-antisense regulation. Okay, so I had these two data sets of cancer-responsive long non-coding RNAs and sense-antisense pairs, and I wanted to ask the question, were any of the long non-coding RNAs in pairs regulated or implicated in both cancers? So what I did is I overlapped the transcripts from the melanoma experiment with the transcripts from the breast cancer experiment. And very interestingly, I found 6 long non-coding RNAs upregulated in cancer compared with controls, which also had a sense-antisense partner that was coordinately regulated. That means that in cancer, either both the long non-coding RNA and the coding gene were up or down. And interestingly, 2 out of these 6 pairs directly included a coding gene, which is either currently in clinical trials as a therapeutic agent or experimentally being tested. So the next thing I wanted to look at is what is the network of these sense-antisense pairs. So in order to impute networks based on the sense-antisense pair, I looked at transcription factors binding in a 1 kb region centered on the TSS of both transcripts. And I also used BioGrid to identify potential protein-protein interactions involving the coding gene of the sense-antisense pair. So so this is the example from this data set that I'm going to continue the rest of this talk focused on. This is choline kinase alpha. It's on the minus strand. This is a shot of UCSC, and this is a schematic of the sense-antisense pair. So the coding gene is here in purple on the minus. The long non-coding RNA, which is again coordinately regulated, upregulated in cancer compared with controls. This is the long non-coding RNA. And the data up here just revisits our melanoma data. These are custom UCSC tracks, which show that both the long non-coding RNA and the coding gene are coordinately upregulated in cancer compared with controls. There's one more thing I'd like to point out. So interestingly enough, there is a two microRNA binding sites directly at the region of overlap between this coding gene and this long non-coding RNA. And I'll discuss more of that later. So I want to take a moment to look at the conservation of this long non-coding RNA. And so this transcript has both primate-specific and conserved features. On the left, you're looking at a sequence alignment of a spliced donor site in the long non-coding RNA. You can see it's conserved only through primates. On the right is the polyase signal site of the same transcript. As you can see, it's conserved through vertebrates. So if we build networks that include long non-coding RNAs, we can introduce not just complexity, but primate-specific complexity. So this is a small network that I drew for choline kinase alpha. These are the protein-protein interactions I got from BioGrid. These are transcription factors upstream. And these are the 2 microRNAs that I showed you earlier. So microRNAs negatively regulate coding genes through a binding site. And one of the interesting things that I noticed when I looked at CHKA was that it interacts with a lot of other coding genes which are known to increase cell growth, proliferation, and replication. So CHKA inhibitors are in stage 1 clinical trials right now as a tumor suppressor. And so because the long non-coding RNA that this gene shares sense-antisense with is also coordinately expressed, it may be possible that if we're looking to therapeutically knock down CHKA, knocking down the long non-coding RNA coordinately could have positive effects. So this is the same network, just with the long non-coding RNA introduced. You can see that because there's that sense-antisense overlap on opposite strands between the long non-coding RNA and the coding gene, it shares these microRNA binding sites. So I've told you that the coding gene is upregulated in cancer. The long non-coding RNA is also upregulated in cancer. One possible reason for this coordinate regulation could be that the long non-coding RNA transcript could have a physical interaction with the mRNA transcript in the cytosol because of the exonic sense-antisense, and this could shield the mRNA transcript from the attenuating effects of the microRNA. This is a hypothesis put forth by Piero Pandolfi called the competing endogenous RNA hypothesis. It promulgates that some long non-coding RNAs in sense-antisense overlaps could be positively regulating their sense-antisense partner through just such attenuation of negative signals. So, just in summary, because R and other labs have shown that many coding genes have long non-coding RNA partners, if they are coordinately regulated, it may be therapeutically advantageous to include long non-coding RNAs in these networks. And future work in this project will include quantifying the co-expression patterns between the sense-antisense pair and quantifying downstream binding targets, and just wet lab validation of overexpression in RNA experiments. So I would like to thank very much, again, the organizers, all the members of my lab, all our collaborators, and our supporters. So I will take questions. Thank you.
Were the MERS acting against the choline kinase A or against the non-coding RNA?
Where are the, when you say they, what?
The MERS, microRNAs.
Oh, so yeah, so microRNAs canonically negatively regulate in the cytoplasm based on an interaction with dicer proteins. Oh, the long non-coding, the microRNA binding site is in the 3' UTR of CHKA, which is at the region of overlap with the long non-coding RNA.
Was it acting on the long non-coding RNA or
the... It will act on the coding gene. Okay, thank you.